Hello student, good afternoon. Today we will start the new topic, that is larval forms of Trematoda. Larval forms of Trematoda. In Trematoda, 5 types of larval forms, 4 to 5. First is Meracidium, second is Sporocyst, third is Redia, and fourth is Cercaria. And sometimes, occasionally, that cercaria develop and transform into metacercaria, insisted metacercaria. But that is the 4 larval stages, is main 4 larval stages of Trematoda. First larva, that is the Meracidium larva. Meracidium larva is the first larval stage of Trematoda. As the Meracidium is formed, the hatching enzymes is secreted in response to exposure to light. The enzyme dissolve the cementing material by which the operculum is attached, thus releasing the operculum. Expansion of the granular cushion accompanied by exosmosis of salts and other materials from within the egg pushes of the operculum. The hatching occurs in water and free swimming Meracidium penetrates the intermediate host. Structure of Meracidium. It is an elongated, about 0.07 millimeter long creature with a broad anterior end and a narrow posterior end. Anteriorly, there is a conical or apical papilla or terebratorium. It is ciliated except in the region of apical papilla. However, anterior cilia are slightly longer. The ectoderm is formed by 21 hexagonal plate-like cells which are arranged in 5 rows. The first row consists of 6 small plates. The second row contains of 6 plates. Third row has 3 plate cells, 4 plates in fourth row and 2 plates in the fifth row. The arrangement of the ciliated plates is quite constant and is of considerable value in determining phylogenetic relationship in Fasciola hepatica. The cells of the first row arranged are 2 dorsal, 2 ventral and 2 lateral in position. The cells of second row are 3 dorsal and 3 ventral. In third row, the cells are 1 dorsal and 2 ventrolateral. The fourth row has 2 right and 2 left cells, and fifth row has 1 left and 1 right cells. In this Meracidium larva, at the anterior side operculum, below the operculum viscous cushion, eye spot, and both the lateral side flame cells. The ectoderm is followed by musculature consisting of outer circular and inner longitudinal muscle fibers. The musculature is followed by a layer of cells forming the subepithelium. The ectoderm, musculature and subepithelium forms the body wall of larva. At the anterior tip of the Meracidium, is found mobile apical papillae, a pair of cephalic glands or penetration glands located in the mesenchyme empty to the outside near the apical papilla. The secretion of these glands aids in dissolving the host is tissue during the penetration process. Beside a sac-like multinucleated apical glands is located between the two cephalic glands. Previously it was supposed to be gut of larva The sensory organs of Meracidium includes two eye spot. Eye contains pigments are situated at the surface of the second row of epidermal plate over the brain. The eyes may be primary receptors involved in the process of hatching. When kept in dark, few Meracidium hatch, but illumination rapidly induces hatching. The brain of the Meracidium is a large cephalic ganglion lying in the parenchyma behind the apical region. It is except from this nerve center. Fibers are oriented in all directions of innervate the various body tissue. There is a pair of flame cells or protonephridia which are excretory organs and their ducts open to the outside between fourth and fifth row of epidermal plate cells. During the differentiation of the Meracidium within the egg cell, certain germinal cells become trapped in the parenchyma. These develop into germ balls by increasing in number and size and becoming envelope, eventually giving rise to the next larva generation. Such germ balls are usually located in the posterior region of the body. Meracidium swims about in water or moisture filled where cattle usually pass out their feces. It dies in 30 hours unless it is able to reach intermediate host, which is always an amphibious snail of the genus Limnia. Intermediate host in a case of Fasciola indica is Limnia acuminatalis, while in the case of Fasciola hepatica, intermediate host in Limnia truncatula. When larva enters the tissue of the intermediate host with the help of apical papilla, it reaches soft parts such as mental, pulmonary sac, and hepatopancreas. Penetration into the intermediate host is helped by the secretion of the penetration glands or other glands. On reaching the intermediate host, ciliated epidermal cells are case of, while eyes and brain are the lost. Larva swells up and changes in the shape and size so as become sac-like structure called the sporocyst. Germ balls of the Meracidium do not disintegrate. Second larval stage is sporocyst. First is Meracidium, second is sporocyst. The sporocyst is an elongated hollow sac-like larva. Its body wall consists of non-cellular cuticle followed by the musculature and subepithelium. The brood chamber is hollow space in the center of the sporocyst, within which are found the germ balls that eventually give rise to the next generation. Sporocyst contains no alimentary canal, nervous system or reproductive system. The protonipedia are present opening laterally by the pores. Outer layer is cuticle, inner layer is muscular radii in the body of sporocyst. Some epithelial cells also, both the lateral side, that is flame cells, are also present. Opening of protonipedia, propagatory cells or the germ cells also present. The sporocyst moves about the muscular contraction in the host tissue, absorbing nourishment from them and causing them a considerable damage. Its germ balls develop into the next larval forms, the radia. Each sporocyst produces 5 to 8 radia. Some of the germinal cells are again set aside within the radia for formation of the next larval forms. Okay, thank you.